Hi, I'm Professor Morris Maduro. I'm a professor of biology in the Department of Molecular Cell and Systems Biology at UC Riverside. I've been here for 22 years. All animals begin as a single cell, the fusion of sperm and egg to make a zygote. And what we're interested in our lab is how that single cell transforms itself into a multicellular animal. We use a model system, a small nematode, to answer these questions, because the nematodes that we work on grow in a very short time scale. They go through 2 generations a week, and a single animal of the species we study, C. elegans, can produce 300 more of itself. And this is a wonderful feature because the very rapid development allows us, and their small size, allow us to watch them under the microscope. We can use all sorts of modern innovations like fluorescent proteins and the ability to edit genes using the procedure of CRISPR to figure out how different genes that come on at different times and places in the early embryo cause an animal to form from a single cell that undergoes many, many cell divisions to become a complete animal. More recently, we've started asking these questions on an evolutionary timescale by taking more distantly related species to C. elegans, which is the species we've studied the most, and asking how those gene networks might have changed over evolutionary time, over tens of millions of years. CRISPR is a genome editing technology that started out as a naturally occurring defense system in bacteria. But geneticists have harnessed the power of CRISPR to make specific changes in the genomes of organisms. This is possible in humans to cure genetic diseases, but we use it in our laboratory as a mechanism of changing genetic information so that our nematodes can tell us what their gene functions do by us going in and mutating those genes so they don't work anymore to see what didn't happen in development. So here's some of the technology that we use to make our magic happen in our research. Over here is a compound microscope equipped with fluorescence. This allows us to watch the development of these worm embryos in real time, and we can visualize fluorescent proteins in living embryos. We can also look at gene expression in fixed embryos. Behind me over here is a microscope that we use to perform microinjections. So we introduce CRISPR mechanism into the gonads of the animals in order to change their genetic information. We also introduce additional genes that help us figure out how these animals develop.